Hi everyone, and welcome back to Unraveling Rare. My name is Eleni Pitiniega from Unravel Biosciences, and we are at the 2025 CSNK2A1 Foundation Conference with...
Hi, my name is Volkan Okur.
Clement Chow.
Heike Rebholz.
Dr. Gabrielle Rushing.
Matt Huentelman from TGen in Phoenix, Arizona.
And Dr. Rushing, what brings you to the conference this year?
So I am the Chief Scientific Officer of the CSNK2A1 Foundation, and this is my first conference.
So I wanted to present my work on the missense variants in CSNK2A1 that were waiting to be identified in other affected individuals. And also being a core member of the OCNDS community, I'd like to be with the family whenever it's possible.
Oh, I have been to, this is my third conference, my third OCNDS conference. So I've been in that space for a while. And yes, I'm working on CK2, I'm working on OCNDS with mouse models. So of course I have to be here.
I'm here to share about our drug repurposing results and the partnership we have with the foundation.
I am a geneticist, so I like to study how genes impact disease. And we have a very long history working in this disease and trying to understand it.
How has your work historically supported the OCNDS community?
So luckily, I was one of the first people, first author who identified this disorder in initial 5 individuals. And I'm a clinician by training and also laboratory geneticist. So I have been trying to identify, first in the first maybe half of the last 10 years, what are the most common symptoms that we see in individuals with OCNDS. And recently, thankfully, now having more researchers on board that are focusing on these different aspects. I'm trying to support the community with what I'm doing on my daily life, molecular diagnostic, analyzing the genetic data, and also making connections with, again, the symptoms and the identified variants in those individuals.
So as you probably know, OCNDS is not just caused by one single mutation, but we have a variety of different mutants. And we're studying different mouse models that represent certain of these mutations that we call hotspot mutations that many patients are affected by. And so we are comparing the behaviors and the biochemical kind of fingerprints of these mice to understand better what these different mutations actually engender.
Yeah, so as a Chief Scientific Officer, I direct our scientific strategy. I collaborate with our clinicians and researchers to make sure that the science that we're doing is sound and really focusing on the issues that matter most to patients. So a little bit of everything.
So we have identified one of the mutations that is associated with this disease and characterized it in some of our patients. And we are very interested in further understanding the differences between different patients. So it turns out that different patients can have different genetic changes, and they also have different problems. Trying to understand that relationship is really important because it could tell us about new therapies.
Are there any findings or ideas that you've heard at the conference so far that have either surprised you, inspired you, or challenged you?
I think the challenge is that there's so many variants in the CSNK2A1 gene, and how everyone is focused on a very small subset of variants, and we really need to understand kind of more about all of them.
Yes, what was super interesting to see is that people who work on different models, like me, the mouse, other people on, like you, or people who have the flies, we come to the same conclusions, and that's very encouraging.
Well, everything that we've heard about here has been really inspiring. But I, in particular, am very interested about some of the new work that's happening in the search for drugs for OCNDS. Now, we know that none of them are perfect yet, but these beginning stages are just so motivating. I think it really gives me a lot of hope, and I hope the families feel that way too.
Yeah, so we did this really unique format earlier today of a crowdsourcing session where we asked families to ask researchers questions and vice versa. And then we were working together to identify the top priorities or gaps in research that mattered most to the families. And what surprised me most today is one of our researchers asked if there were skills that were present in individuals with OCNDS that they were better at than, let's say, a neurotypical child. And a lot of the families spoke up and said that their child's memory is superior to that of their typically developing children, like remembering details from years ago about what they wore on a certain day, for example. And I had no idea that that was a thing. And it really helped me to better understand how we're going to frame looking at that moving forward.
And as a subject expert in this disease, what do you think is currently the biggest mystery in OCNDS?
So OCNDS is like linked to CSNK2A1. It's a gene-based diagnosis actually. And it is a part of a larger unit, CK2. And even before we published our paper, like we knew that this was an ocean, and more, like every day we are learning that how much we don't know about it, and it's really there are a lot of things that we need to identify to understand, like how these variants in affected individuals, disturb the normal functioning of the cells in human body and result in those phenotypes. So we can address those and reverse either the progress of the disease or hold the progress and like give some relief to the individuals.
I think the biggest mystery really comes down to the fact that this particular gene is involved in a protein that does a whole lot of work in our cells. It does a whole lot of different things. And how just that one change can cause all the things that we see in front of our eyes that the patients suffer with. Linking those together is what I think is the biggest challenge.
Still haven't really an understanding of what is the mechanism that brings us to the expression of the disease. We really don't know. We know it's in the brain, but what are the pathways? CK2 is a kinase, so it should really affect somehow the phosphorylation status of proteins in the cell. But which ones are the ones that carry that drive, the phenotype or the symptoms in patients? It's still a very, very big mystery. And I think there are lots of labs working on that question together. So I'm hopeful that we're getting closer, but right now we don't have really any knowledge about the mechanism.
Biggest mystery probably goes back to what the protein itself is doing in cells. So each variant tends to, or appears to date, have a different effect on the function, whether that's where the protein is localized in the cells, what it's phosphorylating, how much the protein is there. And I think that looking at that in a variant-specific manner is going to help us understand whether someone with a specific variant exhibits a certain type of symptom or has a different symptom severity. So I think overall genotype to phenotype correlations are a big unanswered question.
Given your involvement and your critical role in even discovering this disease, how do you feel being at this conference, this OCNDS conference, and then also how do you feel about all the progress that has been made since you discovered the disease?
Oh, it's amazing. So first of all, this is my family. I mean, I was super lucky. This foundation, this gene, these individuals, they gave me a career, satisfaction in what I'm doing, that seeing that I have been able to affect lives in a positive way. That was amazing. And seeing that we have been growing a lot. I know like Jennifer also keeps saying this, like when we first met in the lab meeting room, like 10 years ago, we were like three of us, me, Dr. Chung, Jennifer, and like trying to, imagine like what the future holds and where we could be in the next 5-10 years. And like we only had one researcher, Heike, back then, who was interested. And now you can see that this is a very legit conference. It's growing every day, and being part of it, being able to see like how it's growing, it's a really privilege for me. And again, like I said, this is my identification, like professionally in my scientific and medical world.
What gives you hope when you think about the future of OCNDS?
Well, I look around the room here and see all these really great scientific minds who are trying to tackle this problem. That gives me a lot of hope because I know I'm surrounded by a lot of smart people who are working on this. But also the work that the foundation does, matching up the patients that are dealing with the disease and researchers who are studying it, that is so critical to understand that relationship and to have those relationships as we move forward together. That's really, really exciting.
Yeah, what gives me the most hope is this is probably the most collaborative group of scientists I've ever seen. Normally, when you go to a science conference, there's usually people disagreeing at every corner, and any disagreements that did come up were handled so professionally and kindly. And I think we've really developed or established a huge team of people that are going to work together and continue working together in the future.
I think what gives me hope is groups like mine and yours working together to try to bring a therapeutic option to these kids and these families. I think that's seeing that in action, I think is really promising and really exciting, and it's kind of the right way to move.
Yeah, what makes me hopeful is that there are new people coming into this community, new approaches that us biochemists would never have dreamt of. Like, well, you Unravel, you're coming with a completely different and very difficult to conceptualize, approach, and you get results. And actually, when we look then together with our biochemical findings and your magic algorithm, and there are overlaps, then it gets really exciting. And yeah, that makes me hopeful that something is going to happen.
Being involved in the OCNDS space for as long as you have, how do you feel about coming to the conference, and how do you feel about all the progress that has been made as a community so far?
It's surreal almost. I started planning this conference about 18 months ago, and I think I've said this earlier today, but when we first started planning this conference, I think we booked the hotel contract for about 20 rooms. And our conference, as you can see, has nearly tripled in size, and it's pretty amazing to see how many families and researchers came from across the globe. We have people from France, Germany, and even Australia coming here. So it really gives me hope that so many people are here and committed and passionate about helping.
Finish the following sentence. In the next 10 years, I hope that the OCNDS community will blank.
Have many therapeutic options ranging from drugs to gene therapy, and we'll be one step closer to making helping these kids live healthy, happy lives.
The OCNDS community will be strong, will have tackled symptoms. I think there is not a cure, but they will have tackled some of their most problematic and life-disturbing symptoms that the patients have. I think they will have a better life, the kids, in 10 years.
I hope that the OCNDS community will look back at this moment and say that's where it started. So in 10 years, I hope that there's some reflection to say, look where we are today. We have these new things. I don't know what those are, but new things that are making an impact in this disease. And I hope that the whole community can think back and say, wow, we were really doing something back then.
Feel more confident with not only the current unknowns, right? Feeling more confident in what they can expect, and feeling more confident in that we are approaching a treatment or ideally a cure.